just to talk about membrane proteins. I mean, we are using this as an example of telling you a little bit more about simulations. That's a nice example. We have a nice example of water channel here, for which we have done a lot of simulations. We have learned a lot about the system, and that's exactly the point I'm trying to make. How much you can learn about the system if you analyze your simulations enough. So at the same time, for those of you who might be interested in membrane proteins, you get to know a little bit about lipids and how to set up a membrane protein system. So, so membranes are, of course, important. Separating the two sides is important for the cell, but at the same time, you have to be able to communicate information across the membrane. So yeah, that's an important aspect. So membranes are composed of lipid molecules. And these are very nice sort of molecules because they self-assemble in bilayer like structures. You have a head group that is charged, that faces water, and then you have these hydrophobic tails that aggregate in the middle to form the bilayer structure. So they self-heal if you puncture them, so the ends come together again, and it's a very convenient way of using a barrier between the cytoplasm and the extracellular region. So now, one thing that comes always to mind is the fact that lipid diffusion is rather slow. So if you look at the biochemistry textbook, they describe lateral diffusion of a lipid within the same leaflet. You can see these two lipids are exchanging in this example here. This is described as rapid. And then you have sort of flip-flop motion of lipids going from one membrane to another, one leaflet to another one, described as a very slow process, essentially not happening, because you have, on the average, one happening every several hours. So unless you have an enzyme that does this for you, you don't have flip-flop motion of lipids on a sort of relevant biological time scale. Whereas lateral diffusion happens. But again, if you look at the diffusion constant for this process, lateral diffusion, you're looking at 10 to the minus 8 centimeters squared per second. Just to compare this, water has 3 orders of magnitude faster diffusion constant. Okay, so, but you can get an idea how much water moves in your simulation, and if your water was 1,000 times slower, how much it would move. That gives you the idea how much lipids move. So why am I telling you all this? The relevance of this process is that, yes, I mean, essentially lipids don't move much. You get in 5 microsecond simulation, you might have on the average 15 angstrom displacement of lipids, which means they might exchange a little bit with a couple of neighboring lipids. Essentially, you don't have lipid mixing. And that's important because oftentimes people would like to describe a membrane, a sort of biologically relevant membrane, which is composed of many different kinds of lipids. You have asymmetry between the two leaflets as well. But that's a very difficult process because they don't mix, meaning that you place them somewhere and they're going to stay there probably for the rest of your 200 or 500 nanosecond simulations. That's something to keep in mind. There are ways to deal with this. Coarse grain simulations is one thing, so that allows you mixing of lipids, formation of bilayers, and things of that sort. But again, you lose your atomistic description. And if the details matter, then you can't use coarse graining there. Another way of actually dealing with this problem, again, that's another general technique that you have to keep in mind. You can have multiple simulations. Going back to what I was telling you yesterday, so instead of trying to capture everything in a single simulation, using a very clever way, we can have 10 simulations. Maybe you can place the lipids in it initially differently. Your initial configuration, your initial mix will look different. And then you place your protein in 10 different membranes and run them in parallel. It's going to be even faster on supercomputers. So keep that in mind. Sometimes doing multiple simulation is a really good trick to increase your sampling or solve some of your problems. Yeah?
I've never done MD, so I have a question, but could you assign a different velocity of this group of atoms representing the membrane?
Different velocities.
In order to increase this?
Well, I mean, there is, of course, I mean, initially, I mean, you have exchange of kinetic energy. I mean, it's gonna, initially you might assign higher velocity. I don't think it works because then things move too fast. But even if you can manage making these guys initially faster, they're gonna exchange kinetic energy to other components of the system and equilibrate to the temperature. So that's what you're suggesting can be sort of translated in, maybe we can increase their temperature, the temperature of the system also. But then, I mean, bilayers are not very stable. At higher temperatures, they start falling apart. You have a lot of problems associated with that. So that's, yeah, so that's an interesting idea, but no, unfortunately, that doesn't work in this case. So, okay, so let me actually now, I'm going to be a little bit faster because this is a 4-hour session. I'm trying to squeeze it into 2 hours. So that's actually what happens if you look at an atomistic simulation. Essentially, you can see lipids move up and down very significantly within a few nanoseconds, but you have no exchange between their positions. So that's one reason that usually in most of the simulations you see in the literature, we are using a single type of lipid, PE or PC, for those of you who are interested, are the most popular kind of lipids that we have here because we don't expect to see mixing. That doesn't make sense. Recently, over the last few years, people have started adding cholesterol. Now we have mixtures of PE and PS and other components. So we are getting better. Simulations are getting longer. We started seeing some mixing. We can run multiple simulations. We can start with coarse graining and sort of mix the lipids a little bit and then convert them to atomistic models and then place the protein in the membrane. So things are getting better, but still sort of mixing is a major issue. So, and of course, I mean, Klaus talked about coarse graining. You can coarse grain your system. In the case of lipids and membranes, coarse graining has been very successful. And the reason is, essentially here you have two types of major interactions. You have polar interactions in the head group, and you have nonpolar interactions in the tails. That's the major drive of formation of bilayers, micelles, vesicles, and things of that sort. And the distinction between these two classes of interactions is very nicely preserved in coarse-grained models. So you can actually reduce the number of particles by an order of magnitude. You'll have less particles, and then these particles have a softer kind of softer degrees of freedom because their vibration is going to be bigger, and you don't have to have femtosecond timescales to calculate their trajectories. You can actually go to 10, 20 femtosecond, 25 femtosecond time steps. So that gives you another order of magnitude. So you can have 2, 3 orders of magnitude increase in the time scale of your coarse grain simulations, easily going to tens of microseconds and seeing nice things like formation of a vesicle here. So you can see this is actually one of the original calculations done by coarse grain models by Sivertjan Marink, who is one of the leaders in this field. He's the developer of the Martini force field, in case you're interested. So you can see he starts with sort of distributed lipids in solution, and they sort of form a nice vesicle. So you don't pay attention to this. The time scale here is not real. This is essentially sort of just adding up the time steps. Because, I mean, if you do 100 nanosecond simulation of this process atomistic, there is no way you can see anything like that. So what you're seeing is almost sort of 10 or hundreds of microseconds. So you can get things of that using coarse graining. Yes?
It sort of looked like it was pulsing. So, like, see, it kind of has these jumps.
Oh, I see. That's an
in other directions.
It's sort of like.
Is that an artifact?
That's a very good question, actually. Yeah, so you sort of, it goes there, something like, I never paid attention to this aspect. I don't know.
Sampling of the.
I try to explain it using sort of sampling, but again, I mean, you know, you have 24 frames per second, so it's clearly every half a second or every 1 second, something like this. Seems that they turn back every second, you know. That's interesting. I don't know. I'll think about it. Yeah. Okay, so now let's move to membrane proteins. And again, I mean, the main point I would like to make here is that, so when you're running a biomolecular system, MD simulations, then you end up with a large trajectory file. You have hundreds of thousands of atoms or hundreds of or thousands of groups interacting with each other. You have many, many, many time steps. You have a huge trajectory. And then it's very difficult to know where to start, what to analyze. Running simulation is not a big deal. So hopefully after this workshop, everybody here can run a simulation. But then making discoveries is not trivial. So really analyzing the system is something that helps you a lot. So you have to have some ideas what to analyze. And probably it's very important, as I highlighted here, to get to know your system. So this is very important. So, you know, if you don't care about protein X or Y, it's better to play with a toy system to work with your models and your ideas. But if you're working with a protein, every protein has a different character, has different properties. You're doing it, probably you're interested in certain properties of the protein. You have specific questions. So it's very, very important to know the system. So some of you might have heard this. I usually say it's like, you still constrain the protein. We started with a very sort of bad initial configuration with the membrane, completely ordered, which is not nice. But nowadays you have a better way of doing it. In VMD, you can generate a patch of membrane which is pre-equilibrated. That's a better starting point. But that's the idea. You sort of optimize the alignment of the two parts. Water comes. If you don't have enough water coming with your lipid, you can add more slabs on top using the solvate plugin in VMD. And then when you look at it from the top view initially, so there are gaps here. So that's something to try to minimize, because if you remove too many lipid molecules, then you will have too large gap. It takes the system a little longer to fill those gaps. And of course, your total area is going to be reduced. So it's better to try to preserve as many lipids as you can in the first step. So let me actually go through this. Okay, so let me skip the old one and go to the recipe for membrane protein. I mean, there are many different recipes. So then again, you can do it however you want. Because in this phase, when you're modeling your protein in the membrane, you're still putting the system together. It's not a real thermodynamic ensemble. You can be playful. You can do whatever you want during the modeling phase. Once you have the whole system put together and you want to start your production simulation, then of course you have to be careful. But during this phase, you can do whatever you want. So one way is, of course, we want to have the protein aligned with the z-axis. So that's the sort of membrane insertion. It might tilt a little in the membrane, depending on the lipid environment. Most of the proteins that are deposited in the PDB come in a sort of aligned form. These days, they are aligned with their sort of main axis aligned with the z-axis. So if you place your membrane in the XY plane, which is usually the case, then you're fine. If it's not the case, you can download the aligned version of the protein from the OPM database. It's orientation of proteins in membrane. Thank you. And then we have actually a very cool script called orient, which can be used to align this, to find the principal axis of any object, actually. So this is very good for doing analysis. For example, if you're interested in the angle between two helices, you can find the principal axis for this element and for this element, and then do the dot product to get the angle between the two elements. So you can use this to really do very interesting analysis. You specify a group of atoms, and for that group of atoms, this script calculates the moment of inertia and the principal axis. That's a useful one. Keep that in mind. So then the next step is to decide on the lipid type, what kind of lipid type I want to use, how large the system should be. As far as lipid types is concerned, for those of you who are planning to do membrane protein calculation, usually bacterial systems, we use PE because that's most representative lipid head group in E. coli, which is the bug that is usually used to express the system. If you are using a mammalian protein, PC is the most representative one for which we have good parameters. POPC, DOPC could be your lipid type. Sometimes these mammalian systems have been expressing E. coli to get the crystal structure, for example, so you can also use PE. Sometimes there are experiments done in different lipid bilayers. You know that it works in different lipid environment. That gives you more flexibility in terms of lipid composition of the membrane. So you can use the membrane plugin in VMD. There are many other pre-equilibrated membranes also in the net, on the internet, so you can download a patch from the internet and then use it for your simulation. You want to have actually a large enough patch so essentially, in periodic boundary condition, the protein doesn't see its image in the neighboring image of the unit cell. But if you make your system too large, of course, then your sampling will be limited. So there is always a compromise. But I would say you want to have at least 3 or 4 layers of lipid on each side of your membrane, so the lipids can relax to their sort of relaxed thickness and are not too perturbed by the membrane. Yes?
So can you assign a different composition to the diplet through the membrane builder?
Not yet. I mean, we have an experimental version that allows you to do that, but it's not released yet. So we were just talking about that, so we should probably release the beta version of that. So the idea is that, yes, you can have different lipid compositions for the 2 leaflets. And actually, by the way, what you can do, I forgot, I should add it here. You can use Charm GUI. This is a web interface developed by OnePill that allows you to have really many different compositions. They have many more lipid types, cholesterol and everything. So that's very actually handy and useful. You should use that to generate different compositions of lipids, and it takes care of the area that you need. It's the best tool out there to generate different membranes. So I should add it here, actually. So then overlay. So we talked about this step. We try to maximize the matching between hydrophobic belt of the protein and the hydrophobic core of the membrane. At the same time, we keep an eye on tyrosine and tryptophan rings to adjust the position of the protein in the membrane. And again, I mean, this will be taken care of very effectively by your simulation. But the more you help the system, the faster it reaches an equilibrium, sort of reasonable configuration. Why not doing these initial simple steps to make it faster? Then we remove lipid and water molecules that are overlapping with the protein the same way that you did it for your water box. We try to keep as many lipids as you can, because otherwise you will have a significant shrinking of the membrane patch. If there are sort of small clashes, lipid molecules are very large. You might have only 1 or 2 heavy atoms interacting with your protein or overlapping with your protein, but then you remove the whole 150 atom lipid. That introduces a large gap. Sometimes actually water starts going into those gaps before they get filled. They get filled with lipids. Water has the opportunity to go there. Sometimes they get trapped there, and it's very tedious to solve these problems. So you can do it much more intelligently by maybe if you can look and see there are a few lipids that are minimally overlapping with your protein. Maybe you can use move command with mouse in VMD, just pull them away from the protein. It's going to be fine when you minimize it later. That way you can keep that lipid instead of introducing a big gap there. So then you might need to add water and ions. You can use solvate and auto-ionize at this step. If you have the problem, I mean, water usually doesn't penetrate hydrophobic areas unless they are very large, or unless you have a couple of polar residues in that area. So if you have a problem of water penetrating your membrane, we have a script that prevents water from crossing certain Z planes, XY planes that you specify. So you just contact us. You can use those scripts to keep water out of the membrane during the initial modeling step. Then we sort of, once you have a sort of reasonable starting point for your simulations, then we are going to constrain the protein, because the protein comes from the crystal structure. We want to preserve that while we are doing all these crazy modeling steps. We don't want to introduce perturbation to the protein during this phase. We constrain it, not fix. What is the difference between constraining and fix?
The strength of the spring constant?
Say it again.
The strength of the spring constant, or the potential constant for each potential?
Well, I mean, fix means it's fixed. It doesn't change at all, right? I mean, the position is fixed. But whereas constrained, you constrain it, you ask it to stay around its initial position by applying an additional harmonic potential to the position. So there are, I mean, I'm saying constrain, not fix, for two reasons. I think these are interesting aspects. First of all, I mean, when you fix an atom, it means that its position is not going to change during the simulation. XYZ is going to stay the same. So that means there is no need to calculate forces acting on this one. So essentially, you don't calculate forces between two fixed atoms. You still calculate forces arising from a fixed atom acting on other atoms, but you can expect actually a large part of the system fixed, and that reduces the calculations that you have. So that makes your calculation faster. Whereas if you're doing constraining something, again, if it's few atoms, it doesn't make much difference. But if you're constraining a large part of the system, then you're adding additional terms to your potential energy, these harmonic potentials that you're adding. That makes your calculations more expensive. It makes it slower. So that's from the speed perspective. But from the sort of modeling perspective, what you're doing here, again, okay, so I think I need to back up here. Let me see if I have it here. Okay, so what we are doing here, we are going to melt the lipids, and then later we are going to use constant pressure simulations to allow the system to change its volume and to fill the gaps, to fill the empty gaps that we introduced when we removed the lipids. So we do a constant pressure calculation, meaning that we are asking the program to keep the pressure constant. So the program is going to calculate the pressure. How is the program calculating the pressure? Any idea? How is the pressure calculated in an MD simulation? Any guess? So it uses sort of virial terms, so in which essentially you're looking at interaction between atoms. So if, for example, if atoms are repelling each other, they want to make room, that gives you a sort of high pressure inside the water box. And you need to increase the volume to provide that space that is needed to remove these clashes and repulsion. If atoms are attracting each other, then you have sort of negative pressure inside. The system tries to reduce the volume to bring the pressure up to the level that you have specified. Okay, so we look at interaction between atoms in order to calculate the pressure, and we use that pressure as a target to get the right volume for the simulation system. So if you fix these atoms, the interaction between them will not be calculated. So if you fix the whole protein sitting in the membrane, half of your system is missing from your pressure calculation. So the pressure that you calculate is not going to be correct. So in early days, you might even have actually really, really wrong pressure, and the system tries to adjust the volume too much. You end up with really non-physical kind of description of the system. Yeah, the reason is like because, I mean, the way you change the volume, I mean, is that you are going to scale the position of all of the atoms in the system. So for example, let's say these two atoms are here. You want to change the volume by 10%. You're going to move everything by 10% toward the center of the simulation box. And if this results in a too short bond distance and these atoms are moving, then they can adjust nicely in the next step. But if they are fixed, then that's going to mess up the whole system. Yeah?
So if you're building in a loop and you want to justify the angles for that loop, is it best to restrain when you energy minimize or constrain?
Well, I mean, restrain and constrain, I'm always confused, but I think in terms of NAMD, they both have. Are you talking about the distance or position constraint? Distance. Distance is, I mean, in terms of how you do it, you are going to apply a harmonic potential for that distance. Now, you want to call it constraint or restraint, doesn't matter. They are the same. So the contrast is between constraining and fixing something. Fixing means it doesn't change at all. Constraining means that you are applying a harmonic potential. It is allowed to move around that point, depending on how strong that harmonic potential is, but it can move. That's the contrast. But constraining and restraining, I don't know what's the difference, actually. Yeah. So I would constrain your bond distance. Yes? So the same way that you apply fix, so there is a block in NAMD configuration where you say fix atom on, and then you tell the program what file it should use and what column in the PDB file it should use to flag the atoms that are either fixed, or for the constraint block you do the same thing. So you use usually the occupancy or the B factor column in your PDB file to flag the atoms that need to be fixed or need to be constrained, and then use that PDB to tell NAMD what atoms are fixed or constrained. In the case of constraint, you have to also specify the harmonic force constant that you're using, how strongly you are going to constrain them. Okay, so now, so first again, I mean, the whole idea is that we want to first increase the disorder in the lipids, because that's how they should be. They should be a liquid phase. And we want to preserve the protein structure during the modeling phase. So we keep it constrained. We can apply a very heavy constraint, very strong constraint on the protein atoms, and then allow water, ions, and lipid molecules to move during the initial phase. So once the lipid tails are a little bit more disordered, we call this a sort of melting phase. Then we release everything, minimize. We still constrain the protein heavy atoms, but release everything else, including water. And now we switch to an NPT ensemble, constant number, constant pressure, constant temperature, to allow the size of the system to change in order to pack lipids and water against the protein. Okay, so again, it's not real and a real ensemble in a way, because we are taking advantage of this ensemble in which the volume changes just to do our modeling, just to pack the lipids around the protein. So I have already mentioned this. Watch water molecules. Just make sure they don't penetrate the membrane. They don't get trapped inside between lipids and the protein. They're going to have a hard time to get out. Maybe it doesn't matter much, but that's an artifact. It might cause problem at some point during your simulation. So we can either keep them out, prevent them from penetrating the membrane, or if you have a couple of water molecules in there, you can actually just delete them after this step. But if it's too much, then you should use that script that I mentioned to prevent water from penetrating the membrane. So we monitor the volume of the system. Again, here the system size is changing, but we want to do it to the degree that lipids are packed against the protein. So usually you have a sort of fast drop in the volume or in the extension of the system. You can monitor these files in NAMD. XSC gives you the instantaneous volume of the system or extension and the position of the origin in the system here. And XST has a history of the volume. So you can see X, Y, Z components, and you can see how fast they are changing. So you're monitoring the volume, specifically actually X and Y, which is the size of the membrane patch. You have to have a system that has, what's the command? A flexible cell. You have to turn on flexible cell in this case, because flexible cell allows XYZ dimensions to change independently. So in this particular case, because you have an anisotropic system, every time you have XY shrinking, probably the Z has to expand to allow the water molecules that you have in the system. So you cannot have a system in which X, Y, Z all shrink or all expand at the same time. So you have to have a flexible cell. Just make sure that that's on. Okay, but again, if you are using different force fields other than charm, or if for any reason someone needs to use older versions of charm, we don't want to allow the system size to shrink too much, because we have this general problem that most force fields, including earlier versions of charm, have the tendency to take the lipids to a gel phase, in which you have a very small area per lipid. They are compact too much into each other, and that's not biologically relevant. So we want to prevent that from happening. One way of delaying this or avoiding this to a large degree, at least within hundreds of nanoseconds, is to apply a constant area that prevents the system from further shrinking. Still, you might have too much order in your tail atoms, but at least by fixing the area, you can actually delay the process. Yeah?
Yeah, so initially, like, for example, in the membrane protein tutorial, you have two different solvate procedures, the external program solvate and then the plug-in.
Right.
I'm just wondering, you know, if you could get a water density that's maybe incorrect or too high, and then if you're using flexible cell, that's constant volume, right?
Flexible cell means that, no, it's not constant volume.
Okay, so then your density will...
Yes, exactly. But and now that you mentioned the two solvates, I mean, one solvate which is in VMD, which is very simple. You just have a box, you overlay it with your system, and then remove overlapping water molecules. That's a very easy way to add sort of a box of water or a sphere of water around your system. That's fast, but it just works based on the sort of simple test where you look at all the water molecules in the box, and if they happen to be close to the protein, you just remove them. Whereas the other solvate, which comes from a group in Germany, Grubmüller, we call it Grubmüller solvent because he is the developer there, they actually try to place the water by just looking at different orientation. They try to actually fit water on the crevices. You know, they can pack water much, much better around your protein. They start with the first shell, and then they go around that several times. At the end, you end up with something that looks like an egg, kind of sort of solvated system. That is sort of more rigorous. It takes much longer, of course, several hours sometimes, but it places water much closer to your protein. So I think in your tutorial, you're starting with that kind of solvent to solvate better your protein, but then you're going to remove the part which is going to be in the membrane. You're going to remove those water because the water shouldn't be there, and just keep the water that is hydrating the extra membranous sort of parts of your protein. Then you put the lipid bilayer, and then you use the VMD solvate to have the bulk, essentially. Yeah, so these are, again, I mean, as I said, I mean, I'm just having some of the steps to discuss important things, but we can have a different protocol depending on your system. So we don't want to have over-shrinking, so this might be difficult to judge because here we will have some shrinking. That's something we want to fill the gaps, but we don't want to have too much shrinking because at some point you might start ordering your lipids too much. That's a little bit difficult, but so you can actually monitor the size of the system. If you look at the Z, you can see a sort of, or as XY, I'm sorry, the area, you can see that you have a quick drop and then you have a very slow shrinking with older versions of the charm force field or with other force fields. So this can go on almost forever. So probably this first phase is what you want and this second phase is what you don't want. Maybe at this point we are going to switch to a constant area kind of calculation that keeps the area for you and prevents that over-shrinking phase. So then at this point, we feel that water and lipids are packed nicely around the protein. We're going to release the protein and to go through the minimization and of the whole system if you need it, and then a short NPT simulation because now, if you release the protein, now the protein is also able to move a little bit. Maybe it can be more accommodating of the lipids, sort of absorb them a little bit better. So we want to allow the system size to adjust a little bit. A short NPT simulation, half a nanosecond or so is enough. And then after that, we switch to either a sort of NVT in which the volume doesn't change at all, or you can actually, a better choice would be NPN, which means the pressure is adjusted only along the membrane normal, along the z-axis, and constant area kind of simulation to allow water fluctuation and everything. So that's a better ensemble. So for the recent charm force field, charm 36, you don't have to worry about this. You can continue with NPT, at least for the first microsecond. It's fine. It has been tested for most lipid types that we have. It's doing fine. So you don't have to worry about this over-shrinking problem at all. Why do you need to minimize the system? Why do you need to minimize the system?
To bring it down from the mountain.
To bring it down from the mountain, exactly. Exactly. Absolutely. So what happens if you are on top of the mountain?
You waste your time.
You waste time. No, actually, it's not. So no. What? Could explode. We are getting closer.
But also, the MD is based on the fact that it is independent of the starting point. If you extend the simulation as long as possible, you have to reach the global minimum. So if you are not in the minimum already, you spend more time to get what you can obtain in magic first.
Well, I mean, in a way, you are doing MD because you want to go over those mountains. So maybe it's better to stay on top of the mountain and just go downhill in a different direction. I could argue that way. No, actually, I mean, there's this little explosion. So you see, I mean, MD, you calculate forces. And based on the forces, you are going to move in the next step, right? If you have very large forces on some atoms, they're going to move so much, then they start bump into other atoms. And in the next time step, although you are taking time steps that are very short, 1 femtosecond, but that is enough for them to move so much that they might bump to other atoms, feeling an even larger force that pushes them back, larger force, and that results in instability of your simulation. So for some systems, you don't have to even minimize it. The PDB files have been already minimized because they use actually similar force fields before they deposit the structures. Usually they refine them and relax them. In former times, we had a lot of clashes, but nowadays it's very unlikely to have a very bad protein that has clashes. So it's already sort of relaxed to some degree. But the main point is to have a sort of cool enough system. You don't have any hot spots in your protein, so you can slowly give temperature to it and slowly bring it to a sort of a stable simulation. Still, actually, sometimes you minimize the system. Sometimes you don't even minimize the system. It runs well. Sometimes you minimize it for hundreds of thousands of steps. Still, it doesn't work. So initially, in the first few picoseconds of your simulation, you might see a lot of problems, a lot of crashes, depending on your system. So just be patient. Maybe you can, again, increase the minimization steps. Maybe you can start with a lower temperature. Don't jump all the way to room temperature. Run it maybe at sort of lower temperature. Maybe you can decrease the time step. Instead of 1 femtosecond, maybe you can have 0.5 femtosecond. That prevents the system from moving too much. So there are a few tricks that you can use initially to reach a stable sort of state. Once it runs for a few picoseconds, then it's usually fine and nothing happens to it. But in the first few picoseconds, you might run into a lot of problems. Sometimes you have a problem in your modeling. We have seen actually very interesting cases in which when we place lipids, and then you have a sort of tyrosine like this, and a lipid tail going through the tyrosine like that. So really, we have had cases like this, very interesting. And actually, this is stable, by the way, because the forces balance each other. There is a huge force coming from all sides, but it's happy. It sits there forever, and of course, never goes out. So there are lots of interesting things that can happen during the initial phase. So anyway, so that's what happens after you do all this packing and 1 nanosecond relaxation. You can see that the lipids nicely have packed against the protein. You can see still gaps here. You know what these are? Anybody wants to guess? Side chains, exactly. I'm not showing the side chains. We are just looking at the helices. But it's nicely packed. You can see this is beautiful lipid, disordered, going out. That's nice. And then, as I told you, as far as the thickness of the membrane, we try to optimize this initially when we place the protein, but it's going to be adjusted very fast. In contrast to lateral diffusion of lipids, which is very slow, this sort of adjustment of thickness is very fast. So this is what we got in 1 nanosecond, initially starting with a completely ordered arrangement. But you can see how the lipid bilayer changes its thickness to match the hydrophobic belt of the membrane. So you can see this is very, very nice. So now the first system that we simulated, the crystal structure was done in high concentration of glycerol, which happens to be the substrate of this protein. So in the crystal structure, they had 3 copies of the substrate inside the channel. So one way of looking at it is, this is not physiologically relevant. We don't have 20% glycerol in our body. So let's remove it and just use water and a few glycerols in solution. But then we thought maybe we can take advantage of this artificial kind of condition to our advantage and see if we can use it to describe, for example, glycerol permeation through this channel. So I would like to make another point about the arrangement of this protein. I told you it's a tetramer. So you might think these are 4 independent channels. So maybe I should simulate one of them instead of 4, because the system size is going to be too large if I simulate 4. But in this case, first of all, if you have a monomer, maybe the interaction with the membrane will be different. And second of all, having 4 copies of the protein increases your sampling by a factor of 4. Why not taking advantage of 4 of them at the same time? So you might argue the same way if you're working with other proteins. Sometimes it's better to keep the dimer configuration instead of trying to reduce the system size. It increases your sampling. So we started with this, and then we did a simulation. And this is a very old movie. This is one of the 2 movies that took us a couple of days to make them in a very cold room sitting next to the computer. So scripting everything, and it's I still like it. So you see we have 4 monomers, and then you might see glycerol molecules, these little flies. So moving up and down, we had 4 monomers, each monomer 3 glycerols. So we had 12 copies of the substrate. Some of them are moving up, some of them are moving down, some of them actually getting out of the channel completely. So then by sort of stitching together the position of these, or taking snapshots from different glycerols, we could actually describe what might be a sort of pathway for glycerol to go through the channel. It's sort of obvious, but you can actually have a complete sort of pathway and how it interacts with the channel lining, because we had enough displacement of individual glycerols in 4 identical monomers. That was good, so that's nice. So we put this together. This is not a single trajectory. This is a sort of stitched together movie from different monomers and different time spans. But you can, using this movie, we can actually see how it goes through the, we can describe the entire permeation mechanism, not the time scale, not the energetics. So you can see this is the selectivity filter region where we have this arginine. That's one of those charged residues, by the way, that I mentioned. And you can see it has to be linear to go through this step. Yeah, so that's interesting.
You never saw 3,
Sorry?
You never saw 3 glycerols at once.
We had 3 glycerols. I mean, but I'm just showing the trajectory of one glycerol. In the beginning, the first 1/3 of the animation was a trajectory of glycerol 1 in monomer B. And then I switched to monomer D, where I had motion
No,
of.
I mean, like the X-ray, which had 3 in one channel.
We had 3 initially.
But do they?
No, that's not how it
Three at
functions.
once?
That's not how it functions under physiological conditions. You're right. If you have very high glycerol concentration, then you will get 3 glycerols in the channel. 20% is very high. But under real conditions, it doesn't work like that. But in our simulations, we kept all 3 copies to have sampling of different regions, in the channel. So that was okay, so far nice, but so what? I mean, what did we discover? So then the next thing was analysis. This is one example. So what else can we do? So let's now analyze so the substrate, of course, is interesting. Let's look at the substrate-protein interaction. Like, this is one obvious thing, right? So what we did was to look at hydrogen bonding patterns between the substrate and the protein. We wanted to see what protein side chains are participating in forming hydrogen bonds with the substrate. So this is, the syntax here is not correct. Please don't use that. But this is just an example to show you how easily you can go through all the frames and do all sorts of crazy analysis that you are interested in VMD. So you just set frame, you define a loop that goes from frame 0 to frame 100 in your trajectory. And then you increase the frame number. That means you take the trajectory to the next step, and then do whatever analysis you want to do, save the results in a file or 2 files or 150 files, and then go to the next frame. This is very fast, very efficient, and you can script it very, very nicely, actually. So take advantage of this to do this. These analysis you can do overnight. You can do many of them. Do extensive analysis of your trajectories, please. So in this case, we said, let's go to that frame, and then we define donors and acceptors. So the donors are sort of atoms from the protein, either oxygens or nitrogens from the protein that are within 2 angstrom of glycerol, from hydrogens of the hydroxyl groups of glycerol. And we dump them in a file called list1. We do the same for hydrogen bond acceptors. We dump them in list2. OK, that was one analysis that we did. And then we take those files, lists. We have a lot of names. We unique them to see what residue numbers and what atoms from those residues are involved in hydrogen bonds. So these are hydrogen bond acceptors, all oxygen and nitrogens that accept hydrogen bond from glycerol. And these are hydrogen bond donors. These are residues that donate hydrogen to oxygens of glycerol. Our arginine is sitting here. The NPA guys are here. These asparagens are here. Yeah, so that's obvious. And if you look at here, just a bunch of oxygen and nitrogens. But then you notice something really interesting. I don't know if you want to guess. It goes back to our discussion about topology file yesterday. Do you see any dominant species here.
A lot of carbonyl.
A lot of carbonyl oxygens, exactly. That got us a paper. Just knowing that carbonyl oxygen is named O. O is the name for carbonyls, for backbone carbonyls. So now, I mean, usually side chains are fine. Side chains are there to interact and do crazy things, but backbones are usually involved in intraprotein kind of hydrogen bonding to stabilize the helices, or beta sheets and things up. So when you have a lot of them, then it's good to look at them. So when you look at them, some of them are on the surface, in these loops outside the protein, so they're exposed, they can interact with the glycerol, but many of them are inside this region, which was actually a big surprise. So but then when you look at these carbonyls, then you can explain here. See, these are 8 of those carbonyls. This 1, 2, 3, 4, 5, 6, 7, 8. And with the 2 asparagens in the middle, they form a complete pathway for glycerol to go through. So now we understand why the protein bothers having these non-helical halves in the 2 halves of the proteins. 4 carbonyls here, 4 carbonyls there, because these are essentially the pathway, define the pathway for glycerol permeation. Otherwise, it's very hydrophobic. You just have a trail of carbonyls in the first half, another trail down here, and 2 asparagens in the middle. 4 hydrogen bond acceptors, 2 hydrogen bond donors, and I tell you why later. So that forms the trail for substrate permeation. And now you understand, okay, this is a function of this, and then what stabilizes this unfavorable configuration? Those 2 glutamates that are highly conserved in the whole family in the transmembrane region. They were already in the crystal structure. In fact, actually, this is a picture taken from the crystal structure. It's not from our simulation. They were already there in the crystal structure, but people ignored them or nobody paid attention to them. Now we can explain why. You have these glutamates, 1 glutamate in each end that forms hydrogen bonds with the amino groups just to make sure that those carbonyls are exposed to the other side completely. So that was the discovery we made. So then you can see also the pathway and the mechanism. Here are the asparagens in the middle that I will get to in a bit. So yes, I think the point here is that, yes, so first of all, we need to look at sort of primary sites to which the substrate interacts with, but then don't stay there. You can go beyond that and see what elements in the protein are interacting with primary substrate interaction sites. So that actually took us to a very interesting mechanistically relevant aspect. Yeah. And those are exactly the 2 glutamates, as I told you, that were surprising us in the beginning. If you look at the sequence, this is always a good idea to do some little bioinformatic analysis and see whether the patterns that you are seeing are conserved in the family. In this case, if you look at all these human aquaporins, you can see you have this glutamate conserved in all cases except 2, right? QQ. And you might say, well, I mean, if it's doing such an important job, how come AQP9 and AQP3 don't have it? But if you look 1 residue before, you can see in both cases you have an aspartate 1 residue shorter or sort of in the next position. And aspartate, if you take into account that aspartate is 1 carbon shorter, and this position displacement, it puts it exactly in the same spot as far as the carboxy group is concerned. So you get the effect from this aspartate, essentially, in these cases. So it's going to be the aspartate that is sitting closer, but it's a little shorter, that forms hydrogen bonds with this. So that's also pretty nice. 1 point on the importance of explicit solvent here. Actually, I mean, we had a lot of competition. Glycerol doesn't go through the pore by itself. It is actually accompanied with water. In fact, the pore is filled with water in the absence of glycerol, and glycerol probably has to come with water. So there is a lot of competition between water and glycerol for forming hydrogen bond to those carbonyls and these asparagines. If you don't have explicit water, then essentially you don't see the displacement of glycerol. It gets stuck into inside the pore. So I think the lubricating effect of water for breaking hydrogen bond is very important in this case. And in many other cases, every time you have this hydrogen bonds important, then the effect of water is very important. So that was the arrangement that we saw, and it's very interesting if you compare this to potassium channels, in which we have a similar sort of arrangement of carbonyls. So you see, I mean, for those of you who know the structure, you know, I mean, Klaus was talking about the selectivity filter. So this is a selectivity filter of potassium channels, and you have 4 carbonyls like this in a very similar arrangement to this. And then even there, if you look, you see that there are 2 glutamates behind each stretch that are important for keeping the arrangement of the carbonyls. So we seem to have discovered a very similar arrangement in water channels. So now the next story I want to tell you is about looking at actually things that don't happen in our limited time scale. I told you some of these channels are pure water channels, some of them are glycerol channels. And we wanted to understand why some channels don't conduct glycerol and why some other members of the family do conduct glycerol. So we decided to take 2 proteins, both from E. coli. One of them is pure water channel, one of them is a glycerol channel. And I guess from the picture I'm showing, you can guess the final result already. But the idea was to energetically understand this. So what we did was to look at the permeation of glycerol through a glycerol channel and try to characterize the energetics. And then look at glycerol permeation through a pure water channel and see if we can identify the position of barriers. Why doesn't it go through the pure water channels? So you might expect this to happen. If you run a very, very long simulation, you should be expecting this to happen spontaneously. But you can't have permeation of this big guy through a simple water channel. It doesn't happen. We know from biology that it doesn't happen. But even in this case, where it happens in biology, we can't simulate it long enough with a few copies of glycerol to see full permeation. So we have to do something else. I think Klaus talked about SMD, steered MD. A little bit maybe, but that's the method that we use every time you want to accelerate things a little bit. So you can use the steered MD in addition to forces that you calculate from your force field. You add manually additional forces, external forces. So you can combine these forces very nicely to induce different kinds of molecular phenomena, like you can induce sort of torque and rotation of something. You can separate the 2 ends of the protein. You can unfold the protein. You can pull a substrate out of its binding site and see how it comes out. You can look at permeation of substrates through channels. So a wide variety of molecular events can be accelerated by this method. But of course, I mean, you are doing things a little bit, not a little bit, sort of significantly artificially, right? So, and you have to be careful about what you get. But again, the question is, should I stop even thinking about the problem because I can't do it in my microsecond simulation, or should I still experiment with that using SMD and see what I can learn from that? So I usually use this example of me going outside the door under equilibrium conditions. If you're watching me, how I exit the door, so probably you will be watching me doing this maybe a couple of days, and then the next week I might take the second step, just come back here, and then maybe a few months I just slowly approach here, but then you have to watch me again going back. So this back and forth random walk treatment might take me years to go through the door, right? So, but then if you gently just bias my random fluctuation by a little force, then you prevent some of those back steps, and you might accelerate the process. So that's very good. But if you pull me too fast toward the door from here, then I will go over this chair, over the table, and over these guys. So these are the artificial barriers that you get because my pathway is wrong, right? And you have no idea of the pathway, of course. You don't know whether it's the opening of your protein. You're opening it like this, but maybe it opens in reality like that. That's the minimum energy pathway you don't get. So you have to be careful. You're introducing hysteresis. You worked too much on me unnecessarily. You had to wait for me to find my pathway here, and this gives you a sort of overestimation of barriers and dissipative work that dissipates into the system without really changing the free energy. So, so that's something to keep in mind. We might take artificial barriers, and the barriers, even if you go through the barriers are going to be higher in general. So at the end of the day, what you want to do is to calculate free energy from the work that you do to the system. That's something that you can monitor very nicely in a steered MD, or every time you're applying the force, you can integrate the force along the path and calculate how much work you are doing to the system to induce the transition. And then we know from thermodynamics that this work is going to be, in macroscopic world, this work is going to be always larger or equal to the free energy difference, which is the thermodynamic property you are actually interested in. So this we know from second law of thermodynamics, but then there is an interesting relationship. So we are going from the initial state to a final state, I to F, and during this you have work and heat exchange, and you are interested in the free energy of difference between the two states. So as I told you, on the average, the work is going to be always higher than the free energy difference, right? On the average of work is going to be larger than the free energy difference that you are interested in. If you look at the distribution of the work, one time you pull me hard, one time I'm already moving in that direction, you use less work. And on the average, your work is going to be higher because some of the work is done to the system unnecessarily. But if you look at the exponential average of work, then there is an interesting relationship. You see that exponential average of work matches the exponential free energy difference. And this is because if you look at this exponential average of work is dominated by those trajectories, by those cases in which work was very small, right? This is a negative term here. So those trajectories in which your work was very small will dominate the average of this property. And you will have, in the microscopic world, actually you have cases, you see this is a small part of the tail here, you will have cases in which the work that you did to the system was actually less than the free energy difference because I was already going there by myself. So the work that you did to me is even less than the free energy change. So those rare cases, and that's a key word there, those rare cases are going to dominate the exponential average, and that results at the end to give you the free energy. So that's this sort of going from this inequality to this equality or identity here. So we call it Jarzynski's identity or equality. So if you work with the exponent here. But again, I mean, the point is, as I pointed out, there are these events are very rare. If you run, I don't know, hundreds of trajectories, maybe a few of them will show this behavior. Most of them still use too much work. So in order to get this, you have to have really repeated non-equilibrium experiments or simulations. So then this repeated is essentially sort of indicating that we have still sampling problem, but in a different form. So in principle, you can get equilibrium properties from non-equilibrium simulations, but you have to do many simulations. This has been actually proven experimentally. There was a science paper in which they pulled a piece of DNA very, very fast many times and showed that you can get the free energy difference between the unfolded and folded case within a 0.5 kilocalorie per mole. Very nice. Experimental proof of Jarzynski's identity. So that's nice. So now let's see, we decided to do this. In our simulation, we put the glycerol here somewhere close to the mouth of the pore, and then we apply a force to the center of mass. So that way the force is distributed between all the heavy atoms. Usually you leave alone the hydrogens. Hydrogens are very small and their mass is small. If you apply a little force to them, you might have very large acceleration on them. So usually you can leave them out. So we apply the force to the heavy atoms, to the center of mass here, and then we pull it in this direction. You can actually point your force at one of these heavy atoms to look at the directionality, whether it should be the 1 or the 3rd position here. Then you can look at actually selectivity against different enantiomers and things of that sort. So you are very flexible in terms of applying force. So we apply this force. The question is how much force you need to apply to see the permeation during your limited time scale. How hard you have to pull me to see me leaving the room in 10 minutes and not to stay here for a few days, right? So that depends on how much simulation you can do. You can actually have the constant force if you have an idea about how much force is needed to induce the event, or you have some experiments you want to match. Or if you don't know how much force you need, then you can sort of use a constant velocity simulation, what we call, which is a variable force scheme. So what you do, instead of applying directly force to me, you're going to connect the spring to me and then pull the other end of the spring at a constant velocity, increasing the length of the spring, which increases the force acting on me to the degree that I overcome the barrier. So then after that, when I'm complying, then you wouldn't increase the force unnecessarily. So that way it's a very nice flexible scheme, increasing the force when I'm hitting the barriers. When I pass the barrier, then the force goes down, and it's a better way to avoid too much hysteresis in the system. So then actually you can plot this. For example, here you are looking at the displacement versus time. And you can start seeing features. I mean, clearly the first part is easy to move. And then I hit a barrier here. I spend some time. You have to increase the force here for me to cross the barrier. Then the force goes down again. There is another site here. So these might be binding sites where the glycerol is nicely and happily sitting there, or it could be facing a barrier that you have to overcome. So you can actually look at these spots and see what is happening to glycerol there. You can take some frames from here and here and then run equilibrium simulations there and see what is happening. You can identify interesting regions along the pathway. So through this. Yeah, so the hydrogen binding scheme in different binding sites and things of that. At least you see something happening. Now, how reliable it is, that's a different question. But it can be used to see something. So then we also integrated the force and calculated the work, how much work we are doing to the system to induce the transition. So every time you are calculating free energy, you don't have to go all the way from one state to the other state. You can actually break the transition into many mini steps, and then calculate the free energy for these small steps, and then you can stitch them together to see the overall process. That's a very common practice that we do, including here. So like we started from many different initial conformations or configurations for glycerol and then pulled them a little bit to the left, a little bit to the right, just to get the free energy for this segment. And then at the end, you can put all these segments together to get one continuous profile. So when you do that, yes, this is the free energy we get. So we turn it around to match it with the channel here. Then you can see actually there are large barriers in the selectivity filter where there is a sort of constriction here. And then we have another one after the NPA motif, which is the sort of major binding site. The free energy barrier, the largest free energy barrier we get is about 7 kilocalorie per mole, which is surprisingly good because we had only 4 trajectories here. I almost don't believe it. The statistics is not enough here, but maybe we got lucky. So we get 7 kcal per mole, and experimental estimation of the energy is actually about 10 kilocalorie per mole. So it's nice, you can actually get, but again, I mean, it's just remember that there is a way to connect non-equilibrium simulations to equilibrium properties. So now we are going to repeat this for the system. If you repeat this for the protein that doesn't conduct glycerol, the red curve is what I just showed you for the glycerol channel with a sort of a few kilocalorie per mole barrier. If you repeat the same simulation for the non-conducting channel, then you get huge barriers, 20, 30 kilocalorie per mole here, another 15 kilocalorie per mole here. And then when you look at these spots, then you realize actually that these spots, I think, yes, I have the figure here. Then you look at these spots and you realize that essentially the size of the system, which was a very simple explanation, is too small to allow glycerol to go through it. So this is the radius profile of these pores generated by a program called HOLE, in case you need to do similar things. You just specify a point along the path, and then the program searches for the sort of largest radius, and it draws a sphere at each point. So then you can visualize this nicely in VMD, and you can get an idea about the size of the pore and its height changes as you go along the channel. It also gives you the number. You can plot this, and you can clearly see that the black curve, which is the non-conducting, non-glycerol conducting channel, is much smaller simply than the red curve. So let me actually go to the last part of this, in which we are looking at water. So now we have a few water. I mean, if you remove completely the glycerol molecules, then the channel gets filled in half a nanosecond with water molecules very nicely. So we ran this 4 nanosecond production. If you remember, we had about 18 water molecules going all the way across the membrane. We calculated the conduction rate to be about 1 water per monomer per nanosecond, which matches nicely experiments. So we made a nice movie out of this. So this is the movie that took me 3 days, literally. In the computer room, we had only one computer that was graphically strong enough. Even that computer was slow to do this, and I had to script everything. But it's a nice movie. So, and it made it to the Nobel Prize website. So that's the closest I get to the Nobel Prize. So I am there already. Okay, so that's good. So as you notice, actually, the protein might look rigid, but that's because it was so difficult to regenerate this surface representation. It would take almost half a minute to update this back then. So I decided to show the protein, although everything is moving in the simulation, but when I visualized this and made the movie, then I decided to keep it constant, sort of fixed, only one frame. Fortunately, I have 2 side chains from the protein here, those aspirations that are moving, proving that the protein was flexible during the simulation. But actually, that's what happened. So I'm showing half of the protein only. The front half is removed, so you can see what is happening inside the protein. And one of the water molecules has been highlighted using a different color. That's your heavy water, to see the net translocation of water, sort of. Otherwise, you might just see things moving up and down. You don't realize there is a net translocation. And this is actually a 200 picosecond segment out of that 4 nanoseconds. So you have things going like this, and every now and then, lots of water go down in one monomer. Some of them go up in the other monomer. We don't have any hydrostatic pressure or osmotic pressure across the membrane, so the water exchange should be essentially completely random in both directions. But that's nice, very nice. So nice movie. The water channel got the Nobel Prize. We got it there. But so what? What can we learn from this? And that again, so that brings me to the analysis. two things that you need to do to find interesting things about your system. One is to do extensive analysis. And then when you find something interesting in the behavior that you're analyzing, then you have to look at the molecule and explain it and see what it is and maybe make a nice figure showing that feature. And the other thing is to look at the system. To look at the system very carefully and then see if you identify something interesting, then you quantify that through additional analysis. So really watching the simulation is extremely powerful. So I mean, our eyes, visual cortex is so powerful in seeing a lot of things. You look at me for 1 second and then you can tell the color of my shirt, my thick eyebrows, and what pants I have on and everything. In 1 second you capture all these features. So that's really true. I mean, really watching a simulation in a nice monitor, looking at all those corners that you have memorized based on the crystal structure paper, that can help you really see interesting things. And this is actually something that happened exactly to this project. When we published this paper, we made it to Science. There was another paper in Press in Science. We did almost exactly the same simulation. So we are making a big deal of some feature that I will talk about, whereas the other paper didn't. And the only difference was that we did just more analysis and we saw these features. So what we are doing here, so we are looking at water molecules. So here is we were desperate how to analyze this system. And you see, maybe 90% of the analysis that you do go to your supplementary information or never makes it to your publication. But that's what it takes. You have to analyze it until you find some interesting things. So we are looking at disruption ratio. We are trying to explain why protons are not transported by this water chain. So the disruption could be a reason. If the distance between water molecules is too high, protons cannot easily hop. So we try to quantify the disruption ratio, but unfortunately, the highest disruption you get between neighboring water molecules at any point is 30%. That's not good enough to explain. We looked at the distances. The distances are also fine for proton transport between neighboring water molecules. You can look at the trajectory and see there is a highly correlated motion of all these 5 to 7 water molecules inside the pore. This is time. This is the position along the z-axis. We looked at the correlation. We have a high correlation actually in the selectivity filter, where we have a strict single file, 2 water moves together. In other regions, the channel becomes a little wider. You can even have exchange of water molecules between each other, so the correlation goes down. So we calculated the diffusion constant, one-dimensional diffusion constant. You can look at RMSD, MSD, mean square displacement against time, and get the slope. That gives you the diffusion, which is pretty good, actually. So experimentally, we have a number like that. Remember, TIP3P is not a good water for diffusion. In fact, diffusion constant for TIP3P is twice a real water. But still, our numbers sort of end up being within the experimental estimates of water diffusion. Good so far. One trick that we did here to improve our sampling even further was to break our 4 nanosecond trajectory into smaller pieces. As you note, I don't need more than 300 nanoseconds. Actually, in this case, I only need maybe about a few tens of picoseconds to calculate my diffusion constant. So you can take different segments of the same trajectory and do your analysis and treat them as completely independent trajectories because of ergodicity. So you are allowed to do that. If you have similar kind of analysis, you can take advantage of this trick. Another thing that we did was, and again, I mean, so far what I told you was sort of desperate analysis to find something interesting based on sort of a standard kind of properties. And then we visited our collaborators then we started watching the simulation. And one thing we noticed was that, well, I mean, look, I mean, the water seems to be flipping as it goes through the pore. This was something that we couldn't discover just by doing quantitative analysis, but by just watching the simulation, we noticed, oh, well, what is happening? So water is sort of, if I'm water sort of coming in like this, in the center, it sort of turns and sort of leaves like that. Okay, interesting. Let's analyze it. Let's quantify that, and let's show that it's real. Then we did that by looking at the distribution of hydrogens and oxygens, average over the entire trajectory. Always average it over the entire trajectory, right? So this is another important thing to remember. You might see a snapshot in which a bond distance might be 2 angstrom long, but the next snapshot is going to snap back into the equilibrium condition. You always have you can have very unusual configurations in individual frames, but that's not representative. That's maybe the tail of your fluctuation. So you have to always average the property over many, many frames. So this is the average behavior. You can see from this behavior, you can see in the middle where you have the NPA, the green guy here, you have one water with its oxygen in the center and the hydrogens on the 2 sides. It's like in the middle like this, hydrogens. But then beyond that point, you always have oxygen first, hydrogen next, oxygen first, hydrogen next, oxygen first, hydrogen next, until you hit the bulk, and then opposite orientation on the other side. That is represented by this hydrogen bonding pattern, and this is one of the frames, one of the representative frames showing this configuration. So you see quantifying the effect and showing the representative picture so people can quickly understand what it is. So then we also look at the dipole moment of water along the channel axis, and you can see that clearly it flips at the center. So we're nicely quantifying the effect. Yes?
Could you have model hydronium at the start of the opening of the channel and let it flow?
Yeah, so other people did that. So essentially now let me actually, I will tell you. So what we say that this is a sort of robust configuration of water, which is unusual. Water doesn't like this bipolar configuration. Water likes to be polarized. Either all of the water molecules like this or all of them like that. If you don't have any electrostatic effect from the protein, that's the water chain configuration that you get. So something in the protein is forcing the water configuration to be in this bipolar configuration, and that's something that we also investigated. But so then we took this picture to our collaborators in UCSF, Bob Stroud, who crystallized this, and we showed them, and well, we have something like this. This is interesting. We have never seen anything like that. And then one of the colleagues there said, could this be related to proton transfer? And then I said, well, of course, this is not a very optimal configuration for proton transfer. It doesn't conduct proton. And he said, oh, that's very interesting, because these channels don't conduct protons. We had no idea, because we didn't read the structure paper carefully, by the way, what I'm telling you. So, but that was how we got into science, really by realizing, oh, we can now explain this, knowing a property of the system that can be now explained by this. Of course, the effect is much more complicated. After we published this one, so then let me actually first tell you this. Then they reduced the glycerol concentration in their crystallization medium, and they could get a sort of glycerol-free density for water, for the protein with water. And if you look at these densities, they don't see hydrogens. They can't tell us what is the position of hydrogens and whether they're flipping or not. But the position of oxygens they get here matches very nicely the distribution that we have. So we did that, and actually this hypothesis that so water configuration is important. What is the actual mechanism of proton exclusion? After this paper generated a lot of excitement in the field. Many people placed hydronium in the system. They did QMM calculation or other kind of semi-classical descriptions of proton transfer and identified the position of the barriers. So we have now, it's a combination of a charge at the center. We have a sort of positive field at the center. We have also contribution of desolvation. When you're bringing a charge to the center, you need to have enough hydration effect. Water is polar, but it's not charged. It's easier to bring in a water rather than a proton. As well as the configurational barrier, these are all seem to be the mechanism for proton exclusion. So one last thing actually I would like to point out is that now, so you discover something very nice. If you can verify it experimentally, it would be perfect. But in many cases, we cannot really design an experiment that can address. Like in this case, we had no way for an experiment to tell us whether water is flipping in the center or not. So that's hopefully one day we will have it. But one thing that you can do is to test this within your own simulations. I mean, look at experimental labs. They say, we think this arginine is important for the function. How do they prove it? They mutate that and show it doesn't function, right? You can test your own hypothesis, at least within your simulation setting. So I think that was also very important part of that paper. So you have to remember one of the very strong advantages that we have in our simulation world is that we can play God. We can do anything, right? So easily. You know the PSF file now, go there, change anything. I always use the example of a negative sodium ion. How can you do it experimentally? But we can do it in an experiment. Some people actually made a mistake in the simulation by having a wrong charge, and they see interesting things. Be playful. Really, this is so powerful. Don't start with, OK, I need to have a microsecond simulation of this wild type very carefully. No, I mean, just do a 10 picosecond simulation, pull it apart, see what happens. Really be playful with the system. That is extremely powerful. So take advantage of that. So you experimentalists can mutate side chains. We can mutate backbones. We can do anything, really. So you can put it under sun's temperature. I don't know. I mean, just use your imagination. Really, that's really powerful. So take advantage of it and use it. So now what we did was to say, let's say, OK, this is our wild type behavior. We have the flipping of water. So can we somehow do something to the protein to see if we can neutralize this effect? And that way we can show what part of the protein is responsible for the effect. So then we had a couple of suspects here. Remember, we had these two helices, half helices, and those asparagines in the middle. Helices usually have the macro dipole, so you have a strong electric field at the end of these helices. That might be the reason. And those asparagines, remember I told you have the carbonyls in the two half channels, and then in the middle you have two asparagines, switching from hydrogen bond acceptors to hydrogen bond donors, which makes it easy for the water to actually half turn in the middle and do another turn in the second half. So we had some suspects. So then we decided to, it should be electrostatic, right? Because it's a dipole. So let's go after those guys. And then we decided to neutralize. First, we neutralized the NPAs. How did we neutralize the asparagines? PSF file. Exactly what we did. The simplest. That you go to the PSF file. Asparagine, I don't know, 68 and 200 something, just 0, 0, 0, 0 in the side chain. Done. And then we repeated the simulation. It didn't change much. Then we did it for these half helices. So the macro dipole of the helices come from the backbone, right, from the carbonyl and NH groups. We did the same trick. Go to the PSF, put 0 there, so you don't have the macro dipole, but then something bad happened in the first attempt. So what happens if you neutralize the backbones? The whole helix started to fall apart. Then we used some additional constraints, artificial bonds between carbonyl and NH to keep them in a helical form, but neutral. See, that's experimentally impossible. See? And then when you do that, then you actually start affecting the system a little bit, but not quite. When we combine these two effects together, then you get a nice polarized water chain. So then we show what part of the protein is responsible for the effect. This was so easy to add, really, sort of a couple of 100 picosecond simulations added to the end. But it increased the impact of the paper significantly. Think that way, so don't be happy. What more can I do with testing this? So, OK, I think I'm going to skip the mechanism of proton transport. So you need to have a polarized chain, and we think if this is broken, then it becomes more difficult for So that's we try to make some noise about our mechanism.
Is that from VMD?
No, this part, as you can see, this is a transparent sort of picture laid over the, none of them is VMD actually. There are two pictures, hand-drawn pictures by an artist. So it gives a better impression of what is happening. Okay, questions actually. Yes?
How do you decide to run a long simulation or multiple shorter simulations with the different random?
That's a very good question. I mean, we usually start with, so during the exploration phase, you run a lot of short simulations. But then when you want to publish them, people expect certain time scales. Nowadays you can't go in with a few nanoseconds. Even if you capture the effect in a few nanoseconds, people expect you to run several tens of nanoseconds and at least a couple of simulations of the system. Maybe in one trajectory you see something, but you never see it again. There is a very famous example. Scott Feller had a fellowship at IBM, and they had a lot of computer times there. So he decided to repeat the same 100 nanosecond simulation 26 times. This was rhodopsin and lipid bilayer interaction. So in the first simulation that he did, he saw one lipid going through a sort of hole into the protein. Very excited. He did it 25 times more. Never happened again. So that's something. But of course, I have a different take. So to me, if something happens, I like it because, these are all stochastic phenomena, right? So maybe you were lucky to see them in one simulation. It might be artifact, it might be irrelevant, but we can look at it from a different perspective. Maybe that simulation was the one where you were lucky to see a microsecond process in 10 nanoseconds. There is a probability of seeing these events in short time. So don't discard things because you can't actually reproduce them. But you have to be very careful. If you can actually explain what it is, if you can provide a mechanism, that's another thing. Many people say, oh, I saw a breaking of this hydrogen bond at some point in my simulation. This is great. But they don't explain it, what it means and why it happens. If you have that mechanistic support for the phenomenon that you observe in your simulation, even if you can't reproduce it, you still should be able to report it. But of course, the ideal situation is that you see it happening in most of your simulations. It's a difficult question you're asking. I don't have any recipe. It depends on the system. It depends on the process you want to simulate. If you're interested in the transition between two states, probably I would try to concentrate my computational resources on one, maybe two, rather than trying to do 10 short simulations, because the longer the simulation, the better. But it depends, really. I can't tell you. I can't give you my taste for the particular system that you explained to me. So I would suggest maybe 2 simulations, 50 nanoseconds rather than 10 nanoseconds. But I don't have a sort of formula for that. I usually ask for 10 questions before we go to lunch. So 1 down, 9 more.
I asked about the drawing.
I'm sorry? You asked about the drawing. 2.
You had a slide, was that a CLC? So we're talking about the pseudo-fold symmetry.
Oh, I see. Right. So, okay. So that's.
It
Okay.
looks like a lot like CLC, right?
Well, that's.
Next.
Yes. That's interesting that, again, these are 2 cases of ion channels. And you can see in ion channels, you have in potassium channels, you have the same half helices concentrating the same end, generating a very negative field here, which is optimal for a positive ion. These are potassium channels. In the case of chloride channel, you have these 2 half helices pointing their positive end, forming a negative site, or a sort of site for a negative ion. What we have is we have the same sort of helical elements here doing a similar thing, sort of generating a positive field here that helps with the water reorientation at the center. These are ion channels, these are water channels, but they are using same elements in different configurations.
On the slide where you had the plot from the SMD, where you had like the constant force and constant velocity.
Go ahead.
What is
This?
Right, that one. So on the right plot there, it's constant velocity. What is the y-axis?
The y-axis is z.
With this constant velocity, why isn't it just like a
No,
straight
I said the constant velocity, say it constant velocity because you are pulling the other end of the spring with constant velocity. Whether I follow it or not,
Okay.
it's a different, you might actually, that is a linear.
Okay.
If you plot that, it's going to be fixed. But, so I'm glad you asked this because one thing I wanted to mention is that we usually use a stiff spring, because if you don't use a stiff spring, you might need to stretch this spring 50 angstrom before you have enough force on me to cross the first barrier here, right? And then once I cross the barrier, I don't need any force anymore, right? Because it's flat, but still because you're 50 angstroms away for another 48 angstrom, you keep pulling me with significant force. So it's good to have a stiff spring that stays close to me and I follow it much closer. That is also easier to when you want to put together several trajectories and do averaging and do this kind of things. So we usually use a very stiff spring. If it's too stiff, then your noise is going to be, your force fluctuation is going to be huge. Force fluctuation is usually very high, but it makes it even worse if it's too stiff.
So with this constant velocity, is the dummy atom always sort of relative to the glycerol, or does it have one linear trajectory?
Oh, that's a good. So again, I mean, so you specify a vector. So for example, let's say if you want to pull me from here to there, your vector will be along the z-axis. So if I'm here, your vector moves with me.
So it translates with
Yeah,
it.
yes, it's a vector, not really toward the position. So that way, if your substrate wants to move a little bit like this along the channel because it's not perfectly straight, you can still do that.
Yeah, I have
5 more.
I think it was yesterday's presentation on Monday by House. The simulation of the FM tip on the sphere, the spherical particle.
So it was a viral Casper, right?
So was that steered MD on the tip?
I think so. Right.
So the tip was.
Right. I don't know the details of that, but I think they have a representation for the tip, maybe a bunch of dummy atoms forming a surface or something, and then you can apply the force to the center of mass of those and just push them down. Yeah, it has to be some sort of biased simulation. Right. Yeah. Mm-hmm.
Just say a little bit about treatments of peripheral membrane protein interactions, and are those even practical if you're just using PCEPD membranes?
Okay, that's a very good question. I love that question. Because actually that reminds me, I wanted to show you a couple of slides, and I completely forgot. This counts as 3 questions. So actually this is, peripheral membrane proteins are those proteins that only bind to one surface of the membrane. And here again, the slow lateral diffusion of lipids was one of our main problems. So what we ended up doing was to, again, as one way to increase the diffusion coefficient for the lipids, we decided to completely remove the tails where we don't care essentially about. This is now after 3 years of trying many different things. I'm just summarizing it in 10 seconds. So it's very simple. We just remove this and fill the gap with an organic solvent, which is keeping the sort of energetics more or less the same. So that way you don't have to coarse grain your lipid bilayer. You have the exact representation of the head groups and a few first carbons in the tail. But then the diffusion constant, these are all spontaneous processes that take place. Let me, this was the first simulation that we had to do. We had to pull it down. This is a full membrane. We had to pull it down. Sometimes it pushed the membrane down without going into it. Sometimes it would insert. But then we had to we were not happy with the statistics we had. Then we came up with this idea. It was really a teamwork many different ideas coming from different angles by these guys. And then sort of it's crystallized into this model. We have to calibrate how much of the tail we keep, how much of it we replace with the solvent. If you have more questions about this, I'm glad to talk with you guys in this afternoon. So this is what we claim is the first simulation atomistically in 22 nanoseconds that shows the formation of a bilayer. As I told you, you can do this coarse grain, but you can't do it atomistically. So you start with random position or sort of on a grid distribution of lipids, and they nicely form a bilayer. So you can think, for example, you can have a mixture of different lipids, throw in your protein also, and see how they partition. That's actually the same trajectory. Analysis, you see? Analysis. Just quantify the effect. Then if you look at the distribution of atoms, comparing a full membrane and what we call highly mobile membrane mimetic, the distribution of the head group atoms are very similar. The surface of the protein looks the same, membrane looks the same, and this is the advantage that we get. If you run a membrane, full membrane, traditional membrane, 10 nanoseconds, and look at the top leaflet, for example, and monitor the position of the phosphates, they don't go anywhere, just wiggle around. Whereas this is the distribution of lipids in 1 nanosecond using our model. So you can see how much mixing you start to see. And then that is actually quantifying it here. You can increase actually the diffusion constant by 1 order or 2 orders of magnitude, depending on your setup. And now, things that we had to do with SMD and bias simulations, they happen spontaneously. This is happening in 5 to 40 nanoseconds spontaneously without any bias. And of course, then now we can actually increase the number of sampling. A proud n equal 10 here, like experimental studies. So you can see that the convergence is also very nice. You see, I mean, all of them are placed about 10 angstroms above the membrane, but you can see after diffusing in water in a random manner, remember? Then at the end, they all sort of converge to the exact insertion depth, which is experimentally extremely difficult actually to measure. It's very difficult to know how deep they go into the membrane. So this might be actually a useful membrane. I'm very glad you asked the question. I completely forgot. Membrane for many different things that you might have. We are trying to expand the application of this membrane to transmembrane systems. So you can see, for example, we see insertion of the whole helix into the membrane in 12 nanoseconds. 12 nanoseconds. I mean, maybe this is one of those lucky trajectories. Maybe next time we need 50 nanoseconds or 100 nanoseconds. But for a full representation of the membrane, you never see anything like this unless you reach several microseconds of calculation. So it's a very mobile, very nice. We see lipid mixing. So if you place your protein and maybe certain types of lipids aggregate around your protein, PS, cardiolipin, cholesterol, you can start actually taking advantage of this. That gives you really rapid mixing of the head groups. In the core, we have still some problems. You know, this is liquid, too much entropy. Everybody is happy there. So the free energies are not very good there. But otherwise, it's as far as this part is concerned, it really works like a charm. Any protein we put there nicely binds and everything. So. Oh yeah, so this is this got the cover page of Biophysical Journal in. Do I have it there? Not for this one. This is not published yet. But the model, and you can see that we are using it for many different peripheral proteins, drug partitioning into the membrane, alpha-synuclein, viral fusion peptides, you know, and many different applications. This is in Biophysical Journal, if you need.
The things that are not supposed to go into the membrane go.
Very good question. That's something that we are sort of trying to. So yes, so far we couldn't see anything, so we invested our computational time first on those things that bind. But now the question is that maybe everything binds if you use this model. So let's try. We are actually testing that. Mm-hmm. Yes? Oh, I see. No, sure. We are using a similar approach to do vesicles and micelles. as well. So you can fill the interior with the liquid essentially, and just have sort of these head groups on the surface of it. Our main interest is really lipid mixing and lipid aggregation and lipid interaction with the protein. So mostly for coagulation proteins, because they bind to the surface of the membrane in a PS-dependent manner, and people love to know where is the PS binding site. Maybe we can design a drug for that spot and have a better anticoagulation drug. So that was the main motivation behind all this. But now we are doing cytochrome P450, how deep it goes to the membrane, how it recruits drugs from the membrane, aggregation of things, absolutely.
So have you quantified the number of flips from leaflet to another?
Oh, this one, I mean, we don't have leaflet exchange.
Well, we still have the low dielectric barrier, right? The organic liquid in the middle replaces the text.
No, it is slow, but it never happens. I mean, several hours in a normal membrane. And even here, in this case, you have still this charged
So you
head
do
group.
account
No.
for
It might, some of them might fly into the water because these are surfactants. You have a small partitioning of these molecules into the solvent. Maybe through the solvent they can go to the other leaflet, but not through the membrane. There was one question, yes?
Do you see establishment of a GUI Chapman layer or things like that?
So, I mean, we did some, we looked at dipolar potential of the membrane, so we are doing fine there, water distribution and everything is fine, but we haven't got there yet. So again, I mean, I see this point, I mean, many people ask me about all the properties of the membrane from this guy. So here, really, our main intention to have a fast enough exchange of lipids to have instead of placing a protein somewhere where it's going to be stuck, usually it doesn't move out or in. Instead of doing that way, we can use this one to get a sort of reasonably inserted initial configuration, and then we grow the lipids into a full membrane and equilibrate that. So that sort of accelerates the initial modeling phase that you have to do manually. But now, of course, we are also looking at free energies, looking at the electrostatic, and see if we can use it in sort of more situations. But we haven't done anything like that yet. I don't expect electrostatic to be a problem, actually. So again, based on the dipolar potential, we are off by maybe 10%. So we should be fine. But the main problem is here. We have this solvent, which is a little bit polar. That's the only way you can have a liquid small molecule. Otherwise, it's going to be gas. Or you have to use a big molecule there. And another problem is entropy. Really, I mean, when you calculate free energies, you see up to this point it's perfect, and then here, especially for large, bulky things, are so happy, you know, they can flip around, and they're so that's yes.
So I guess unrelated to this, but for coarse graining simulations, I guess I'm wondering how do the fluctuation dissipation terms change when you have coarse graining and you're looking at spheres rather than sort of smaller spheres connected.
I see. I don't think. I mean, I don't think the harmonic terms should not be affected. I mean, because your potential, and it depends on your potential energy. I mean, how many degrees of freedom you have in the system, right? I have to admit, I never thought about this. I mean, we don't do much coarse grain in my group, but I mean, you still have to have a sort of equipartitioning between different degrees of freedom, so equally between your harmonic degrees of freedom. We have the same bonds, bond angles. Are you specifically asking for any particular term? Or
Well,
it should have a very similar behavior, because let's say you might treat them exactly as atoms. There is no difference fundamentally. They just have bigger masses and bigger sizes. They're sort of from a fundamental perspective, there should be no difference between these big atoms and the small atoms that we have, right? We have the same kind of potential energy and functional forms for the potential energy. I don't expect any difference, actually. Yeah. Okay, I kept you way too long, so thank you very much for your questions. I will be around until 4 or so, and I have to run to the airport, catch my flight, hopefully. Otherwise, I see you tomorrow again, which is fine. So let me know if you have more questions.